Dear students, now I'm going to discuss the topic polyacrylamide gel electrophoresis, and next I will discuss about Western blot, which can also be called as immunoblot. Now, what is actually the principle of SDS page, means sodium dodecyl sulfate polyacrylamide gel electrophoresis? This particular technique generally used to separate proteins from its mixture by the utilization of an electric field. Here, the proteins that are going to means we are going for separation or uh, trying to separate maybe a particular protein from a mixture of proteins. So first, we have to impart one charge, and anonymously this charge will distribute at the surface of the proteins, at the polypeptides, and all the proteins should be at the surface of the protein should be a similar charge, and they will then be subjected to an electric field. And due to having this particular charge, here the electric field will be applied through a liquid or a semi-solid medium, and there will be two electrodes, anode and cathode. So you can have migration uh, when the protein mixture will be subjected to an electric field. So when electric field will be applied, the molecules flow at a towards a particular direction against or towards a particular electrode. And here the rate of flow of different protein molecules will be different, and this particular difference depends on the basis of their molecular size. So the rate of migration or rate of flow of movement of the native protein through this semi-solid medium that is actually functioning here as a sieving medium is therefore more a reflection of their relative compactness and less an accurate measure of molecular weight. So here we can also calculate their molecular weight uh, from this study. So denaturation of the protein nullifies structural effects on mobility, allowing separation on a true charge by mass ratio basis. So we cannot say that due to having a different configuration or a different degree of structure, so the protein has migrated less or the protein has migrated more. So we cannot say like that, because before going to subject the proteins into the electric field, so first we are denaturing all the proteins. So the proteins now will behave as a primary structure or simply just like a polypeptide chain. So here we can nullify the structural effect, the structural aspect that is actually hindering, that may actually hinder mobility of the protein molecule. So it also separates subunits in multimeric proteins, allowing analysis of large complex aggregates. So this particular type of electrophoresis can be called as one-dimensional gel electrophoresis. So SDS fate can also be termed as one-dimensional gel electrophoresis. Here, this particular molecule SDS, sodium dodecyl sulfate, This is actually an anionic detergent which is used here to denature the protein by breaking the disulfide bridge bond or disulfide bond and gives negative charge to each protein in proportion to its mass. So here in the picture you can see that on the left hand side this is the folded protein structure with intrinsic charges. Here, after application of this anionic detergent SDS, this is actually breaking the intramolecular disulfide bridge bond, and now this is just like a straight polypeptide chain, and throughout the surface they bears a particular charge, that is negative charge. So with SGS, different proteins with similar molecular weights would migrate differently due to differences in folding, as differences in folding patterns would cause some proteins to better fit through the gel matrix than others. So STS is functioning here. It is actually linearizes the proteins so that they may be separated strictly by molecular weight. Now, the STS binds to the protein in a ratio of approximately 1.4 gram of STS per 1 gram of giving an approximately uniform mass is to charge ratio for most protein, so that the distance of migration through the gel, that is polyacrylamide gel, can be assumed to be directly related to only the size of the protein. So no other parameters will come into play. Here only the size matters, size of the protein matters. Now, proteins may be further treated with a reducing agent that is called DTT or dithiothiotol or TRP, that is tributyl phosphine, to break any reformed disulfide bond and then alkylated with iodoacetamide to prevent the reformation of disulfide bond. So this one of the molecule we can use either DTT or TRP. So that is to ensure that no more disulfide bond reformation took place. And another molecule that is actually a tracking dye we are going to use. This is known as bromophenol blue. So it should be added to the protein solution to track the progress of the protein solution through the gel during electrophoretic run. Now, here what is polyacrylamide? Polyacrylamide is a cross-linked uh, uh, cross-linked of polymer of acrylamide, and it will form the gel due to formation or polymerization of its monomer, and it's present in very small amount in prime methylene bisacrylamide. And bisacrylamide, this is a two acrylamide linked by a methylene group. So the polymerization of acrylamide is an example for free radical catalysis. So the polymerization occurs between acrylamide and bisacrylamide in presence of two molecules. One is ammonium persulfate, APS. Here you can see persulfate ion, and the second one is timide. This is N prime tetramethyl tetramethylene diamine, that is actually catalyzing the polymerization between acrylamide and bisacrylamide. So due to here this polymerization is occurring by the production of free radical by ammonium persulfate. So this polyacrylamide is function here as a gel. So why the question will come, why polyacrylamide will be used for this purpose? Because polyacrylamide will act as a gel or sieving agent. It is chemically inert, it is electrically neutral, it is hydrophilic, and very transparent medium for optical analysis or optical detection. So there is no more haziness. So you can detect whatever the migration has happened via this electric field. So that is why these are the four criteria. On that basis, scientists have chosen polyacrylamide for the preparation of gel for separation of proteins. So this polyacrylamide gel is something different with DNA agarose gel, because agarose gel that is vertical, that is horizontal gel, and here it is a vertical type of gel. So protein will migrate from top to bottom. There the DNA will migrate from one end to another end, and it is a horizontal gel. So here you can see the one more difference is there. There in case of agarose gel electrophoresis, throughout the gel, the total percentage of agarose will be same. But here in case of STS phage, the percentage of phage will be different. So the upper part, that is actually known as a stacking gel, and the lower part of the gel, that is known as a running gel. So there you can see there is a difference in the percentage of polyacrylamide gel. So in stacking gel, you can see is a large pore polyacrylamide gel, and here the percentage of polyacrylamide is 4 to 6%. And also here the pH is less. This is 0.6, it is 6.8. So the gel is prepared by using a particular buffer having pH 6.8, and this buffer is known as Tris HCl buffer. So in case of resolving gel, you can see this is small polyacrylamide gel where we have to use 8 to 12.5% acrylamide monomer for gel formation, and it is typically prepared using the buffer, same buffer, Tris HCl, but the pH is basic, this is 8.8. So we can see 2-unit pH difference between the stacking gel and the resolving gel or the running gel. So in case of stacking gel, the gel is cast over the resolving gel. So you have to put first the resolving gel to a certain level, a certain height, whether it is a certain height, and then you have to pour the stacking gel. So the amount of stacking gel will be less, the amount of running gel or resolving gel will be more. So the gel is cast, stacking gel is cast over the resolving gel. The height of the stacking gel region is always maintained more than double of the height of the and the volume of the sample to be applied. So first you have to check the cum size. Then on that basis you have to go for preparation of stacking gel or stacked gel. Next one is resolving gel. So the resolving gel, the macromolecules will say why it is called resolving gel or running gel. This particular gel having the small pore size, it is actually responsible for macromolecule or responsible for separation of the macromolecule according to their size. So resolving gels have an optimal range of separation that is based on the percent of monomer present in the polymerization reaction. So proteins are separated on the basis of charge by mass ratio and their molecular size, and this particular phenomena is known as molecular sieving done by the polyacrylamide gel. So this is a native gel. It's a different type of gel. There are no denaturant is used. Only intrinsic charge and hydrodynamic diameter is used as a parameter for the separation of this particular type of gel. I'm not going into detail because it is not in your syllabus. So here is the total picture of STS phage, how you have to go for first. Maybe say you are going to isolate some protein from a bacterial membrane. So you have to first go prepare the membrane fragments. Then you have to add sodium rhodocyl sulfate, and then you can pour it by through this in this into this well required amount, and same amount you have to pour for all the wells. Then you have to attach the power source with the gel apparatus. Before that, you have to pour some buffer here, running buffer here. Then this is your cathode, this side is your cathode, and this side is your anode. So the protein will migrate from the cathode to the anode, or negative to the positive, because yes, SDS will impart negative color to the all the proteins or the protein fragments. Now, after some time, what you will see migration of proteins, and this bromophenol blue dye, it is actually used here as a tracking dye, will tell you how much your protein has migrated. As the bromophenol blue has low molecular weight, so it will migrate more, and the migration of bromophenol blue will tell you how much your protein has migrated. Then when the total migration is near about 3/4, then you have to stop this electric field and then take it out, and you can go for staining. Here, two type of staining can be used. If the concentration of protein is very less, then you have to go for silver staining, and if the protein concentration is more, then you can stain it using bromophenol blue. To visualize the protein band, here you can see the protein bands will appear like that. Here is the marker. Here the protein band will appear like that, or else you can see here like this. Now here you can see Kumasi brilliant blue can be used for visualization purpose. If the concentration of protein is nearly 100 nanogram, if the concentration is very less, zero point 0.1 to 1 nanogram of protein present there in your sample, then you have to go with silver staining, because silver staining process is more sensitive rather than Kumasi brilliant blue. So in case, what are the advantages and disadvantages? The advantage and the disadvantages are that here in case of SDS phase, the migration is pushing to the molecular weight, so you can determine molecular weight directly. And high, very sensitive, separates 2% differences in mass of the proteins. Requires small amount of protein and stable chemically cross-linked gel. So these are the advantages, but the disadvantage is that the resolution is very poor. Band resolution is very poor due to high alkaline operating pH. Acrylamide gel, it is a potent neurotoxin. So use of acrylamide gel as a chemical, you have to be very much cautious. Gel preparation is difficult and require longer time. And definitely the cost is high because of different chemicals you have to use. And this portion I'm not going. Native gel and SDS page already we have discussed. We are not using any detergent. Simply the separation is based upon the charge size and set. In case of SDS, separation is based upon only the molecular weight, not on the basis of charge size and set. So this is a basic difference between native gel and SDS page gel. These are the applications of SDS page to measure molecular weight, peptide mapping, molecular weight of different kind of protein, estimation of protein size, to determine different protein subunits or aggregation structure, protein purity check, protein quantification, protein integrity, as well as protein size or peptide size you can check. Next, that is in your syllabus, but before Western blot, you should know what is SDS page, because Western blot is the second step of SDS page. First, you have to run SDS page, then you have to go for Western blot. So when you have run your gel, so you have your protein over the within the gel matrix. Now, what you have to do? Then you have to cut particular type of paper. It may be nitrocellulose membrane, it may be PVDF membrane. So you have to cut a required amount of paper, PVDF or nitrocellulose. Then you have to keep it in buffer for few minutes. After that, you have to set up the experiment. Here, you need to sponge a leaf. First, you have to keep a sponge. Over that, you have to keep some blotting paper, pre-wet blotting paper, pre-wet in the buffer solution. Then you have to keep your gel. Over that, you have to keep the paper. It may be nitrocellulose membrane or the membrane or PVDF. Then again, blotting paper and then the sponge, and you have to put the clip on. Then you have to immerse it into the transfer apparatus, fill the transfer buffer, fill with transfer buffer, and then we'll go for constant uh, current. So you have to go for electrophoresis. Then what will happen? Current will pass from the cathode to anode. It will pass through. So this particular technique is widely used for the detection of specific protein in a specific sample, cell lysate, cell culture, supernatant body fluid, blood sample, etc. etc. So here you can see. So this is a sponge, then filter paper, then gel, your SDS bed gel, and then PVDF membrane, again filter paper, just then sponge, and then you have to put the lid on. So at the bottom this is negative, at the top this is positive. So then you have to go for electrophoresis. After your electrophoresis is over, means what will happen? So your protein will migrate from negative to positive. So your protein is there in the gel. So when it will migrate, it will go from gel to the membrane, PVDM membrane. Now you have to mark the membrane and then immerse the membrane in a particular antibody solution, and you have to incubate the primary antibody solution, and you have to incubate it at 4 degrees centigrade overnight. So after overnight incubation, then you have to go for rigorous washing, and after washing is over, then you have to incubate your membrane uh, again with a secondary antibody tagged with a fluorophore enzyme, and this incubation will be at the room temperature for 2 hours. After that, you can go for BCIP NBT, or you can go for chemiluminescence assay. Then you can visualize your protein band over your PVDF membrane like this picture D. So here actually the mechanism is that if you are using horse radius peroxidase, so in this process BCIP NBT, so you have to use luminol. So luminol is oxidized in presence of horse radius peroxidase, and you have to add H2O2. So this will form an excited state product of 3-amino phthalate. So this 3-amino phthalate emits light at 4 to 5 nanometer as it decays to the ground state. So this light should be captured and it should be visualized here. So here is a schematic representation of how this separation is going on. So denature sample with sodium todecyl sulfate, then run the gel from negative to positive, cathode to anode. Then, so on the basis of decreasing size, it will separate in 3 different bands. So the smaller one, middle size, and the larger one will migrate like that. So it will migrate only because of this sieving property of the gel. from negative to positive. Then we have performed this. These are the cassette and sponge. So you have, this is your wet tank transfer apparatus, and you have to go for transfer. So this is your semi dry apparatus. So any of the apparatus you can use. It's a dry, it's a wet, and it's a semi dry kind. So any of the apparatus you can use upon availability. So here you can see that few things you have to take in mind, because if it is a protein and you are a protein, you are isolated the protein from, say, membrane, mitochondrial membrane, or say cell membrane. So primary antibody, this antibody is very specific. Perhaps an immune cell culture is exposed to some protein of interest. So the dilution of primary antibody, generally 0.5 to 5 microgram per ml, should be incubated with the membrane under gentle agitation for 30 minutes to overnight at 4 degrees centigrade. Then the solution is composed of buffered saline solution with a percentage of detergent. Tritonex 100 or Twin 20 can be used to generate porosity. And you have to use either BSA, 5% BSA, or skimmed milk, powder milk you can use as a blocking agent. So before adding secondary antibody, so this should be blocked, the other part, and your primary antibody will be free. Now your secondary antibody, after rinsing the membrane to remove unbound primary antibody, the membrane is exposed to another antibody that is a species-specific antibody, secondary antibody to primary antibody. The secondary antibody is usually linked to biotin or maybe some reporter enzyme, phosphatase peroxidase, such as alkaline phosphatase, phosphatase peroxidase, etc., etc. So this means that several secondary antibodies will bind to only the primary antibody, not with the other portion of the membrane, and enhance signal. So this chemiluminescence method can be used to get the idea or to get the band present, protein band present in your PVGF membrane. So this can be this is the overall mechanism of chemiluminescence assay. Here you can see these are the two compound, and this will develop some transition state, and from there they will convert into a product and will emit some light. Here you can see in presence of H2O2, luminol and peroxide is, if you excite it at 4 to 5 nanometer, then it is showing some green, some blue fluorescence. So here also you can get some fluorescence, and your band will be there. You can detect your band while the protein is present there. So this particular technique can be applied in case of DNA hybridization technique. So Western blotting, actually immunoblotting techniques for forensic laboratories for food analysis. So these are wide application in clinical to food to forensic sciences. That's all. This is actually the reaction structure, how luminol is reacting in presence of peroxide and is emitting some light, means fluorescence, generating fluorescence. Thank you.